Okay, hello and welcome back to the Mcat stream. Well, I have a tuft of hair just going all over the place, but whatever. Anime fan ahoges, rise up. Ugh, can't do anything about that. Unless I use some gel or something. I'm not gonna do that while I'm at home though. Alright, let's get back right into it. We're gonna be starting a bit of genetics now, which is excellent because I love genetics. You can see how much I love genetics. Oh boy. Slight sarcasm. Genetics is fine. Let's see if we can get this to be even more gamer of a science. Oh my god.
Not until the structure of DNA was established by Watson and Crick. And their work was based on the work of many others, especially folks like Rosalind Franklin, who essentially provided the bulk of the data for Watson and Crick's work, Maurice Wilkins, and many other folks. It was really
Rosalind Franklin, shout out again. Rosalind Franklin, let's go.
DNA was discovered in the mid-1800s. It was this molecule that was inside of nuclei of cells. And for some time, people thought, oh, maybe this could be a molecular basis of inheritance. You can imagine what you would need to be a molecular basis of inheritance. It would have to be a molecule or a series of molecules that could contain information that could be replicated, that could be expressed in some way. But it wasn't until 1953 when this double helix structure of DNA was established that people said, hey, this looks like our molecule. So first, let's just talk about
this is our guy, guys. It's a zipper. It's gotta be it.
About why the structure lends itself well to something that stores information, that can replicate its information, and that can
Replication.
in future videos. So this structure right over here,
And the fact that you can split it down the middle, replicate it. Man, that's convenient. Copy and paste.
You have these rungs, and these rungs are actually where the information, the genetic information, is, I should say, stored in some way. Because these rungs, it's a sequence of different bases. When I say bases, wait, this says acid. Why are you saying bases right over here? Well, the word deoxyribonucleic acid comes from the fact that this backbone is made up of a combination of sugar and phosphate. And the sugar that makes up the backbone is deoxyribose, so that's essentially the D in DNA. And then the phosphate group is acidic, and that's where you get the acid part of it. And nucleic acid, this was found in nuclei.
Volume is a bit low on my end for the Khan Academy vid.
Also, it is actually mildly acidic all in total, but for every acid, it actually also has a base, and that base, those bases form the rung of the ladders. And actually, each rung is a pair of bases. And as I said, that's where the information is actually stored. What am I talking about? Well, let me talk about the 4 different bases that make up the rungs of a DNA molecule.
Gattaca moment.
Adenine. And so, for example, this part right over here, this section of that rung might be adenine. Maybe this right over here is adenine. This right over here. Remember, each of these rungs are made up by, it's a pair of bases. And that might be adenine. Maybe this is adenine. And I can stop there. I'll do a little more adenine. Maybe that's adenine right over there. And adenine always pairs with the base thymine. So adenine pairs with thymine. So if that's an adenine there, then this is going to be a thymine. If this is an adenine, then this is going to be a thymine. Or if I drew the thymine first, well, I'll say, okay, it's going to pair with the adenine. So this is going to be a thymine right over here. This is going to be a thymine. If I were to draw this, it would be a thymine right over here. Now, the other 2 bases, you have cytosine, which pairs with guanine, or guanine that pairs with cytosine.
Alright, so pretty basic stuff, but definitely want to write this down. Want to make sure that we got our bases covered. Literally. Bases.
So actually, if this is, let's say there's some cytosine there, let's say cytosine right over here, maybe this is cytosine, maybe this is cytosine, maybe this is cytosine, and maybe this is cytosine, then it always pairs with guanine. So this is guanine then, and this will be guanine, this is guanine, this is guanine. I actually didn't draw stuff here, but this is guanine. I didn't say what these could be, but these would be made of pairs of, they could be adenine thymine pairs, and could be adenine on either side, or thymine on either side, and they could be made of guanine cytosine pairs, where the guanine or the cytosine is on either side. Actually, just to make it a little bit more complete, let me just color in the rungs here as best as I can. So those are guanine, so they're going to pair with cytosine, pair with cytosine, pair with cytosine. And when it's drawn this way, you might start to see how this is essentially a code. The order of which the bases I guess the order in which we have the sequence of these bases essentially encode the information.
Call them base pairs. Here.
People say nature, it's literally genetic, and that's an ongoing debate. But it does code for things like your hair color. When you see that your smile is your parents, it is because that information to a large degree is encoded genetically. It affects a lot of what makes you, and actually not even just within a species, but also across species. What humans have more genetic material in common with other humans than they do with, say, a plant. But all living creatures as we know them have genetic information. This is the basis by which they are passing down their actual traits. Now, you might be saying, well, how much genetic information does a human being have? And the number will either disappoint you or you might find it mind-boggling. The human genome, and every species has a different number of base pairs, to a large degree correlated with how complex they are, although not always. But the human genome has 6 million. Sorry, not 6 million, 6 billion. 6 million would be disappointing. Even billion might be disappointing. 6 billion base pairs. 6 billion base pairs. And when you have your full complement of chromosomes, and this is in most of the cells in your body, outside of your sex cells, the sperm or the egg cells, this is going to be spread over 46 chromosomes. 46 chromosomes, or I guess you could say 23 pair of chromosomes. If you divide 6 billion by 46, you get a little over, on average, 100 million, I think it's 100 and something million base pairs per chromosome. And some chromosomes are longer, actually, the longest are over 200 million, and some might be shorter. That's just on average. Now, this number might, to some of you, might be exciting. You're like, oh, I thought I was a simple creature. I didn't know I was this complex. 6 billion, that's a lot of base pairs. That feels like a lot of information. For others of you, it might not feel so great. You might say, hey, wait, I could store this much information on a modern thumb drive or on a hard disk. I thought I was more unique than that. And of course, we all are special and unique. And I say, I was 6 billion base pairs. I thought I was infinitely complex and whatever else. And there's some arguments for that along some other.
Infinitely complex. Oh boy.
Of the actual human genome. When we talk about
physics. Hello.
in much more depth, imagine taking this zoomed in thing that you have right over here, and over here, I don't know how many we have, like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19. We have about 20 base pairs depicted here. Imagine if you had about 200 million of these base pairs, and then you were to take this thing and you were to kind of coil it up into that thing is a chromosome. Is a chromosome. And you're saying, wait, I have that much information in most of the cells of my body. This thing must be incredibly compact packed. And if you said that,
It's a good thing that these lecturers speak a little slowly. That this 2 times speed actually works really well. Wow.
But just have an appreciation of
Damn.
on why the structure lends itself to being able to replicate the information, or even to be able to translate or express the information. Let's think about if you were to take this ladder and you were to just kind of split all the base pairs, so you just have one half of them. So you essentially have half of the ladder. And so if you only have half of the ladder, you're able to construct the other half of the ladder. Let's take an example. Let's say, and I'll just use the first letter to abbreviate for each of these bases. Let's say you have some. Let's say this is the sugar phosphate backbone right over here. So this could be one of the sides. Let's say there's some adenine. Actually, the right color. So you got some adenine, maybe some adenine right over here. Maybe there's an adenine there. Maybe you have some thymine, maybe thymine right over here, and you have some guanine, and then let's say you have some cytosine, and you have some cytosine. So with just half of this ladder, I guess you could say, you're able to construct the other half. And that's actually how DNA replicates. This ladder splits, and then each of those 2 halves of that ladder are able to construct versions of the other half, or versions of the other half are able to be constructed on top of that half. So how does that happen? Well, it's based on how these bases pair. Adenine always pairs with thymine if we're talking about DNA. So if you have an A there, you're gonna have a T on this end, T on this end, T's right all over here, T right over there. If you have a T on that end, you're gonna have an A right over there, A. If you have a G, a guanine on this side, you're gonna have a cytosine on the other side. Cytosine.
Yes, that is why they are base pairs.
That gives you an appreciation of how DNA can replicate itself. And as we'll see also, how this information can be translated to other forms of either related molecules, but eventually to proteins. And just to kind of round out this video, to get a real visual sense of what the DNA molecule looks like, or I guess a different visual depiction from this, I found this animated GIF that, if you haven't fully digested.
Oh my god, he said GIF.
And you see your sugar phosphate.
What?
The sugars and phosphate.
Feel so betrayed right now.
And then the rungs of the ladder are the.
It's GIF.
So this is one of the bases. That's its corresponding, I guess, partner. And you can see that along all the way up and down this molecule. Very exciting.
It is very exciting. Hey. Alright. Let's talk about how these things actually connect together like Legos.
Have an overview video of
wow,
DNA.
that's so much louder.
You to watch
whoa,
first.
okay.
But what I want to do in this video is dig a
Hello.
little bit deeper, actually get into the molecular structure of DNA.
Chill.
Starting point, let's just remind ourselves what DNA stands for.
I'm the streamer here.
In different colors.
Come on, man.
It stands for deoxyribonucleic acid. Ribonucleic acid. So I'm just going to put this on the side, and now let's actually look at the molecular structure and how it relates to this actual name, deoxyribonucleic acid. So DNA is just a general term for nucleic acid, and it's the term
hella H bonds. That's what I see right now.
It's found in the nucleus of eukaryotes. So that's where the nucleic comes from.
Oh my god.
We'll talk about in a second why it's called an acid, but I'll wait on that. And now each DNA molecule is made up.
This man's voice is nearly as smooth as mine. Goddamn.
So what we call nucleotides. So it's made up of nucleotides. So what does a nucleotide look like? Well, what I have right over here is I have 2 strands. I've zoomed in 2 strands of DNA. I've zoomed in 2 strands of DNA. So you can view this side right over here as one of the I guess you could say the backbones of one side of the ladder. This is the other side of the ladder. And then each of these bridges, and I will talk about what molecules these are. These are kind of the rungs of a ladder. And a nucleotide. Let me separate off a nucleotide. So a nucleotide would. So what I am cordoning off right over here could be considered a nucleotide. So that's one nucleotide, and it's connected to another. It's connected to another nucleotide right over here. And on the right hand side, we have a nucleotide. We have a nucleotide right over there, and then actually I want to do it slightly different. We have a nucleotide right over here on the right side, and then right below that we have another nucleotide. So depicted here, we essentially have 4 nucleotides. These 2 are on this left side of the ladder. These 2 are on the right side of the ladder. Now let's think about the different pieces of that nucleotide. So the one thing that might jump out at you is we have these phosphate groups. So this is a phosphate group right over here. This is a phosphate group right over here. Each of these nucleotides have a phosphate group. So this is a phosphate group over here, and this is a phosphate group over here. Now the phosphate groups are actually what make DNA, or actually what make nucleic acid an acid. And you might say, wait, the way you've drawn it, Sal, you have a negative charge. Something with a negative charge would attract protons. It would stop up protons. How can you call this an acid? This actually looks more basic. And the reason why this DNA is typically drawn with these negative charges here is it's so acidic, and if you put it into a neutral solution, it's actually going to lose its hydrogens. Actually, the DNA, if you actually want want to be formal about it, the DNA molecules will actually have its phosphates protonated like this, but it so
Hmm.
badly wants to lose these hydrogen protons. So typically would be, let me draw it like this. Let me get rid of the negative charge just on this one. Whoops. Just on this phosphate group over here. So if you get rid of the negative charge, and if this is bonded to a hydrogen, this so badly wants to grab these electrons. So this oxygen can grab these electrons, and then this hydrogen will just be grabbed by another water molecule.
When you're so acidic that you get represented as a base.
But it would be very acidic. As soon as you put it into a neutral solution, it's going to lose those hydrogens. So the phosphates
Oh boy.
are what make it an acid. But it's confusing sometimes because usually when you see it depicted, you see
A OChem arrow though.
it has already lost its hydrogen proton. You're actually depicting the conjugate base here. But
Move those electrons, baby.
It's protonated, or it gets in its acid form, it's protonated, but it readily loses it, and so that's why that's where it gets the name acid form from. So each of these nucleotides, they have a phosphate group. Now the next thing you might notice is this group right over here.
Next thing you might notice?
And it looks an awful lot like a sugar, and that's because it is a sugar. So this
Nice.
sugar is based on, it's a 5-carbon sugar. What I have depicted here, this sugar, this is ribose. So this sugar right over here is ribose. This is when it's just as a straight chain, and like many sugars, it can take a cyclical form. It can take many different cyclical forms. But the one that's most typically described is when you have them. Let me number the carbons because carbon numbering is important when we talk about DNA. If we start at the carbonyl group right over here, we call that the 1 carbon or the 1' carbon. 1', 2', 3', 4', and 5'. That's the 5' carbon. And so you form the cyclical form of ribose. Is if you have the oxygen right over here on the 4' carbon. It uses one of its lone pairs. To form a bond with the 1', carbon. And I drew it that way because it kind of does bend. The whole molecule is going to have to bend that way to form this structure. And then when it forms that bond, the carbon can let go of one of these double bonds, and then the oxygen can use that. The oxygen can use those electrons to go grab a hydrogen proton from someplace to nab onto a hydrogen proton. So when it does that, you're in this form. And this form, just to be clear of what we're talking about, this is the 1' carbon, 1', 2', 3', 4', and 5' carbon. And where we see this bond, this is a 1' carbon. It was part of a carbonyl. Now it lets go of one of those double bonds so that this oxygen can form a bond with a hydrogen proton. So let go of a double bond there so that this could form a bond with a hydrogen proton. So this hydrogen proton is that hydrogen proton right over there. And this green bond that gets formed between the 4' carbon or between the oxygen that's attached to the 4' carbon and the 1' carbon, that's this bond right over here. This oxygen is that oxygen right there. Notice this oxygen is bound to the 4' carbon, and now it's also bound to
Bro, is it bad that I low-key want to take OChem again? Just so that it can go click, click, click. Look, there goes the double bond.
So you form this cyclical form. And the cyclical form right over here is very close to what we see in a DNA molecule. It's actually exactly what we would see in an RNA molecule, in ribonucleic acid. And so what do we think we're talking about when we say deoxyribonucleic acid? Well, you can start with, you have a ribose here, but if we got rid of one of the oxygen groups, in particular if we just got rid of one of the oxygens, we replace a hydroxyl with just a hydrogen, well, then you're going to have deoxyribose. And you see that over here. This 5-member ring, you have 4 carbons right over here. It looks just like this. The hydrogens are implicit in the carbons. We've seen this multiple times. The carbons are at where these lines intersect, or I guess at the edges, and also where these lines end right over there. But you see, this does not have an OH. This molecule, if we compare these two molecules over here, we see that this guy has an OH, and this guy implicitly just has an OH and an H. This guy implicitly has just 2 hydrogens over here. So he's missing an oxygen. So this is deoxyribose. So deoxyribose doesn't have this oxygen. It does not have the oxygen on the 2' carbon. So this,
Of course.
this is deoxyribose. So let me circle that. So what we're
Okay.
this thing right over here, that is deoxyribose.
I'm sure I've heard that. I've heard this explanation like a billion times before, but always nice to remember. Nah, always nice to review.
Right over here, and these we call nitrogenous bases. So nitrogenous bases.
Helps out because I really need to simplify everything in my head.
Nitrogenous base. This right over here is another different nitrogenous base.
Let's see. Maybe I can do.
Has 2 rings. And we have different names for these nitrogenous bases. The ones with 2 rings, the general categorization, we call them purines. So nitrogenous bases, if you have 2 rings, we call them purines. That's a general classification term. Let me make sure. Purines. And if you have
No. I'll just cheat and take a picture of this and then place it in the thing.
We call these pyrimidines. And these 2 on the right, these 2 purines, this one up here, this is adenine. And we talk about how they
It requires way too much visual information for me to represent on the mind map.
And then over here, this single ring nitrogenous base, which makes it a pyrimidine, this is thymine. This right over here is thymine. And then last but not least, if we're talking about DNA, when we go to RNA, we're also going to talk about uracil. But when we talk about DNA, this one over here is cytosine. Cytosine. And you can see the way it's structured. The thymine is attracted to adenine. It bonds with adenine, and cytosine bonds with guanine. So how are they bonding? Well, the way that these nitrogenous bases form the rungs of the ladder, they're drawn to each other. This is our good old friend hydrogen bonds. And this all comes out of the fact that nitrogen is quite electronegative. So when nitrogen is bound to a hydrogen, you're going to have a partially negative charge at the nitrogen. Let me do this in green. You're going to have a partial negative charge at the nitrogen and a partially positive charge at the hydrogen. And then oxygen we've always talked about as being electronegative, so it has a partial negative charge. So the partial negative charge of this oxygen is going to be attracted to the partial positive charge of this hydrogen, and so you're going to have a hydrogen bond. And the same thing is going to happen between this hydrogen, which is its electrons are being hogged by this nitrogen, and this nitrogen which itself hogs electrons. So that forms a hydrogen bond. And then down here you have a hydrogen that has a partially positive charge because its electrons are being hogged, and then you have this oxygen with
Hella H bonds. Let's go.
Same thing between this nitrogen and that hydrogen, and same thing between this oxygen and that hydrogen. And that's why cytosine and guanine pair up, and that's why thymine and adenine pair up. And we talk about that as well in the overview video of DNA.
Ah, yes, an overview. Oh my god, that entire 13 minutes was just on this one figure. God damn, it's a pretty big figure. Okay.
And that's why cytosine and guanine pair up, and that's why thymine and adenine pair up. And we talk about that as well in the overview video of DNA.
My laptop is working so hard to like try and make this work. Wanted to screenshot the thing. Oh my god.
Adenine pair. We talk about. So that forms a hydrogen bond. And then down here, you have a hydrogen that has a partially positive charge because its electrons are being hogged. And then you have this oxygen with a partially negative charge. They're going to be attracted to each other. That's a hydrogen bond. Same thing between this nitrogen and that hydrogen. And same thing between this oxygen and that hydrogen. And that's why cytosine and guanine pair up. And that's why thymine and adenine pair up.
Got it.
As well in the overview video of DNA.
See if I can just paste this onto the thing. Ah, yes, I would like to crop this image. I would like to, however, that's impossible. It contains literally everything that was said. Oh boy. Anti-parallel time.
On the molecular structure of DNA, we saw that DNA typically made up of two strands, where the backbone of each of the strands is made up of phosphate, alternating between just in different colors, a phosphate group, and then you have a sugar. You have a phosphate group, and then you have a sugar, and then you have a sugar, and then you have a phosphate group, and then you have a sugar. And so I could draw the strand something like this. So phosphate, and then we have a sugar. Whoops, let me just draw all the phosphates ahead of time. So you have the phosphates on that end, and then you have the sugars. And you see the same thing on the other strand as well, where we have phosphate with a sugar, then another phosphate, then a sugar, then another phosphate. Let me circle the sugars as well. So you have a sugar there, and then you have the sugar there as well. So on the other strand, it's also going to look like this. So let me draw the phosphates. I'm just abstracting them now. So the phosphate, and then you have the sugars in between the phosphates. And what links them, you can think of them as the rungs on the ladder. These are the complementary nitrogenous bases. And the reason why we call them nitrogenous bases, I actually forgot to talk about in the last videos, is that these nitrogens are really electronegative, and they can take up more hydrogen protons. They have an extra lone pair. The nitrogens have an extra lone pair that can be used up under the right conditions to potentially sop up more hydrogen protons. Now, a lot of people ask, well, if you have these nitrogenous bases here, why is DNA called an acid? Why is it called an acid? Well, the first thing is that the basic properties of the nitrogenous base are offset to a good degree based on the fact that they're able to hydrogen bond with each other. And that's what actually forms these rungs, the rungs of the ladder, when these complementary nitrogenous bases form these hydrogen bonds with each other. But even more, the reason why we call it an acid is the phosphate groups, when they're protonated, are acids. Now, the reason why we tend to draw them deprotonated is when they're so acidic that if you put them in a neutral solution, they're going to be deprotonated. So this is the form that you're more likely to find it in the nucleus of an actual cell once it's already deprotonated. But in general, phosphate groups are considered acidic. And if I were to draw kind of a more pure phosphate group, and I talked about this already in the last video, I would have it protonated, and so I wouldn't draw that negative charge like that. So that's just a review of last time. But let's actually let me just, since I already started abstracting it, let's abstract it further. So let's draw the nitrogenous bases a little bit. So I have thymine here, and I will do thymine in this green color. So this right over there is thymine. So this is attached to thymine, and the complementary nitrogenous base to thymine is adenine. Which I will do.
Okay. Nucleic. Okay. Hang on.
So this is attached to thymine. I have thymine here. I would have it protonated, and so I wouldn't draw that negative charge like that. So that's just that you're more likely to find it in the nucleus of an actual cell once it's already deprotonated. But in general, phosphate groups are considered acidic. And if I were to draw kind of a more pure phosphate group, and I talked about this already in the last video, I would have it protonated. And the reason why we tend to draw them deprotonated is when they're so as the rungs on the ladder. These are the complementary nitrogenous bases.
Okay, wait a minute. So these are actually just referring to the nitrogenous bases, whereas the nucleotide themselves are... I always thought that the nucleotide, like adenine, thymine, guanine, and cytosine, were referring to the entire nucleotide, including the base pairs plus the phosphate group and sugars. I guess that's not the case? Oh boy. Feels like it's just semantics, but it sounds like I just missed something important. Nucleotide. Oh boy. There you go. There you go. Okay, I think that makes a lot more sense now. So hopefully I'm back on track.
and they have these hydrogen bonds between them right over here because they have partially negative and positive charges on either end that are attracted to each other. And then we go to this rung, one rung below it, and what is going on? Well, let's see. We have, I really am running out of colors here. We have this nitrogenous base is cytosine. This nitrogenous base right over here is cytosine. This nitrogenous base here is cytosine, and it is paired up with guanine. It is paired up with guanine. The guanine in this color. So it is paired up with guanine. Right over there. And we even saw this in the introductory video to DNA. Now, you might say, oh, look, these two strands seem parallel to each other, and in some ways that is true, but there might be another interesting thing that you might have noticed is the direction in which they are oriented. I guess is the best way to phrase it. And you especially see that when you focus in on the sugars. Notice the sugars over here, the deoxyriboses, or the parts of the nucleotide that come from deoxyribose. You see the oxygens on the top of the ribose, on the top of these 5-member rings. The oxygen is on top. Well, on this side, the oxygen is on the bottom. And so they're actually in different orientations. Here the oxygen is pointing up, here the oxygen is pointing down. And to get a little bit
anti-parallel,
we can number the carbons on the ribose to think about the directions and use those numbers of the carbons to describe the different directions. So let's number our carbons. So when ribose, so this is ribose, these are both ribose. We saw that in the molecular structure of DNA videos. When we're talking about DNA, we're talking about deoxyribose. So instead of having a hydroxyl group on the number 2 carbon, it just has a hydrogen. So instead of having a hydroxyl group on the number 2 carbon, it just has a hydrogen. But let's actually number them. So this is the 1' carbon starting at the carbonyl group. Let me do that in a different color. So this is the 1' carbon, and I'm just numbering them starting at the carbonyl group. 1', 2', 3', 4', 5'. And then when you look at it as a ring, this was the 1', this is the 2', this is the 3', this is the 4', this is the 5'. Or if you were to number them on this diagram right over here, actually in the DNA molecule, this is the 1', this is the 2' carbon, this is the 3' carbon, this is the 4' carbon, and this is the 5' carbon. And so one way to think about it is, we go phosphate group, and it's connected with what we call phosphodiester linkages. Phosphodiester linkages, that's what's essentially allowing these backbones to link up. But we're going from phosphate to 5' carbon and then through the sugar, we go to the 3' carbon, then we go to another phosphate, then we go to the 5'.
Hey, okay, so I always got confused about 3' and 5'. I feel like this is so much clearer. Honestly, I should have just watched this entire Khan Academy stuff back when I was actually taking these classes, because that would have made things so much clearer. Well, for now, I would like to go back. A screenshot of that thing there in the corner. Oh no, not again.
3' carbon, then we go to another phosphate, then we go to the 5' carbon. Let me label that. This is the 5' carbon, then we go to the 3' carbon. And that just comes straight out of just numbering these, starting with the carbon that was the number 1 carbon, which in a straight chain form, it's at the carbonyl, it's part of the carbonyl group. Well, you see we're going from 5, we go phosphate, 5', 3', phosphate, 5', 3', phosphate. So one way to describe the orientation is saying, hey, we're going in the direction from 5' to 3'. So we could say that we're going from 5', to 3' that way on the left-hand chain. And what are we doing on the right-hand chain? Well, let's number them again. So this is the 1' carbon. Now this thing relative to this is upside down. It's inverted. So 1', 2', 3', 4', 5'.
Okay.
I can do it up here. 1' carbon, 2' carbon, 3' carbon, 4' carbon, 5' carbon. Here you're going from phosphate, 3', 5', phosphate, 3', 5', phosphate. So the way that the sugars are oriented, if you're going from top to bottom, the way we're looking here, you're going from 3' to 5'. So on the right-hand side, it's 3', 5'. And so if you wanted to draw an arrow from 5' to 3', you could look at it like that. And so you could say these are parallel, but since they are essentially oriented they're pointing in different directions, even though they're actually parallel, we would call the structure of DNA anti-parallel. So this would be an anti-parallel structure of DNA. So these two strands, they're complementary. They're defined by each other. The thymine bonds with the adenine, the cytosine bonds with the guanine, or they're attracted to each other through these hydrogen bonds. But the two backbones, they're pointed in different directions. And another interesting thing to think about, since we're talking about the molecular structure of DNA, is how do these things form? How do these things know to orient in this way? And part of what plays part of that role is the fact that these phosphate groups are negative. So these things that have outright negative charge, they're going to try to get as far away from each other as possible. And then when they just keep kind of orienting, getting far away from each other. These are long. These are very, 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 very long molecules. In the introductory video to DNA, we talk about how long these chromosomes are, how many base pairs we actually have. And these are long molecules. All of these phosphate groups on either strand, they want to get away from each other. And then these things want to get close to each other because of the hydrogen bonds. So that's what helps form this actual ladder structure. So DNA, fascinating molecule. We could speak for days about it. It's actually mind blowing when you think about its implications for who we are. But hopefully this gives you a better sense of what it is molecularly. I cannot say it. Molecularly.
Molecularly. Oh boy, telomeres. Where am I going to put that?
Here is a pair of chromosomes as they would appear during mitosis, and the ends of chromosomes are capped with an area known as telomeres.
Alright, so we're getting into chromosomes now. I can probably branch this off of DNA. Yikes. Why is it a squiggly line type? I don't understand. Why must they hurt me in this way? Okay. There we go. Alright. Let's talk chromosomes.
And telomeres are mainly found in eukaryotic chromosomes because usually prokaryotes just have one circular chromosome, so it doesn't have any ends. And what do telomeres do? Well, they protect chromosomes, or protect the ends of chromosomes from deterioration. Why would the ends of chromosomes deteriorate? So the enzymes that replicate chromosomes are not able to get to the very, very end of the chromosome. So they're going to get to, let's say, this area. There's going to be a small spot over here that's not replicated. And since the telomeres don't have any genes in them, that's not really harmful, it doesn't really matter. So what would happen if there were no telomeres? Well, let's take a look at the other chromosome. If there were no telomeres, and let's say the chromosome was only replicated till about here, there would be this area with useful genes that wouldn't be replicated, and that would be pretty problematic. So basically, telomeres act as a buffer zone because they do not contain any important genes. Another thing that telomeres do is they prevent chromosomes from sticking to each other. If chromosomes stuck to each other, then a lot of the genes would be scrambled and genes wouldn't be where they're supposed to be, and that would be pretty problematic. And here's actually a picture of human chromosomes where the telomeres are highlighted in this fluorescence. So you can see the telomeres over here. So you can see at both ends of each chromatid, there are telomeres. And so what happens is that with each time the chromosomes replicate, the telomeres get a little bit shorter and shorter and shorter. So there's an enzyme known as telomerase. And telomerase is able to lengthen telomeres and bring them back to their original length. So there are some cells that replicate a lot and they have a lot of telomerase, so the cell can keep on replicating and replicating. But then there are other cells that do not have a lot of telomerase, and when the telomeres are basically non-existent anymore because the chromosomes replicated many, many times, let's just get rid of the telomeres. So the chromosomes will actually not be able to replicate, and so the cell will not divide again, and it will kind of die. Now that we're talking about telomeres, I want to bring up a topic that's tangentially related, and that is single copy DNA.
Okay, hold up. Whenever we mention enzymes, I always imagine that they're going to break down the thing, but it lengthens telomeres. Okay. Thus, we need to put this and give it a blue supporting role on the telomere. Okay.
Repeat itself, so it might be flanked like other DNA sequences, as opposed to repetitive DNA, which is when you have a DNA sequence that keeps repeating itself. So you might have an ATCC, and then again, ATCC, ATCC, etc. So what's the difference between single copy DNA and repetitive DNA? So here we have a spectrum. On the left, we have single copy DNA. In the middle, we have DNA that's somewhat repetitive. And on the right, we have highly repetitive DNA. So single copy DNA holds most of the organisms, which need an apostrophe there, important genetic information. So basically, most of the important genes are going to be single copy. So since the important genes are single copy DNA is transcribed and translated, and it has a low mutation rate, which is a good thing because, of course, we don't want there to be mutations in the important genes. Repetitive DNA, or DNA that's somewhat repetitive, is found, well, at least in mammals and in insects, near the centromeres. If you recall, the centromeres are the center of the chromatid, or when you have chromatids that are duplicated, the chromatids are attached by the centromere, by that middle part in the chromosome, and they may contain genes that are transcribed and translated, but then there might also be parts of the repetitive DNA that don't contain genes, and those parts are not transcribed and translated, and repetitive DNA has a higher mutation rate than single copy DNA. Now let's take a look at DNA that's highly repetitive. So it contains no genes, and because it contains no genes, it is not transcribed and not translated. And highly repetitive DNA has an even higher rate of mutation than DNA that's somewhat repetitive. So there's lots of highly repetitive DNA that we're not exactly sure what its purpose is. Scientists are currently trying to figure out what the purpose of this highly repetitive DNA is, but there are some sections of highly repetitive DNA that we do know what their purpose is. For example, telomeres. Telomeres are sections of highly repetitive DNA, and as we explained before, their purpose is to basically act as a buffer zone for the important part of the chromosome. And in fact,
It's a really famous example of a highly repetitive
and in human chromosomes, the telomeres are made up of approximately 2,000 repeats of this DNA.
piece of DNA. I forget exactly what it was. I feel like it's like Huntington's, where there's like a very specific repetitive sequence that gets either too long. I think it gets too long or something, and then it really messes with your protein expression. Let's see. Single copy, repetitive. I guess we can put that here, so like single copy. Copy and repetitive. All right, and we're golden. Oh boy, leading and lagging. Not too complicated, but kind of kind of a situation where I don't really like find it super intuitive for whatever reason. Okay. Oh boy. Very small mic apparently. I can't hear it. What is this guy saying right now? Oh my god. I just looked at OBS, it's so quiet.
So this side of the ladder, you could say, it is going in the, draw the line here. This is going in
Oh no.
3' to 5' direction. So this end is 3'.
The volume mixing is
3' to 5'. Notice 3
is not it right now.
5' connects to a 3', and it connects, then you go to the 5' connects to a phosphate. Now on this end, as it says, anti-parallel. It's parallel, but it's oriented the other way. So this is the 3', this is the 5', this is the 3', this is the 5'. And so this is just what we're talking about when we talk about the anti-parallel structure. These two backbones, these two strands are parallel to each other, but they're oriented in opposite directions. So this is the 3' end, and this is the 5' end. And this is going to be really important for understanding replication because the DNA polymerase, the things that's adding more and more nucleotides to grow a DNA strand, it can only add nucleotides on the 3' end. So if we were talking about this right over here, we would only be able to add going that way. We wouldn't be able to add going that way. So one way to think about it is you can only add nucleotides on the 3' end, or you can only extend DNA going from 5' to 3'. If you're only adding on the 3' end, then you're going from the 5' to the 3' direction. You can't go from the 3' to the 5' direction. You can't continue to add on the 5' side using polymerase. So what am I talking about with polymerase? Well, let's look at this diagram right over here that really gives us an overview of all of the different actors. So here is just our DNA strand, and you can imagine it's just somewhat natural in its natural unreplicated form. And you can see we've labeled here the 3' and the 5' ends, and you could follow one of these backbones. This 3', if you follow it all the way over here, it goes, this is the corresponding 5' end. So this and this are the same strand, and this one, if you follow it along, if you go all the way over here, is the same strand. So this is the 3' end and 3' end of it, and then this is the 5' end of it. Now the first thing.
Okay. So what I see right now is a very long laundry list of like, oh boy, okay. Let me see here. I'm gonna start with topoisomerase. Straightening out. Helicase, splitting it. And then, this is where the fun starts. We need to split it in two to represent the leading and lagging strands. So let me see if I can somehow put some room into this diagram here. Maybe I'll swap the sugar around over to this side in the phosphate group. Okay, problem solved. We are good to go. Let's center that a little bit more. Okay, so off of the helicase, we know that there's a leading and a lagging strand. Okay, so leading strand. And then lagging strand. And then we're going to include Okazaki fragments in the lagging strand because DNA polymerase can only go in a specific direction, the direction of which that I always mix it up because whenever people explain things to me with DNA polymerase, they're always like, okay, it's going from 5' to 3', but actually it's going from 3' to 5'. All right, let's think about this. So it's starting on the template strands 3', going to template strands 5', and creating a new strand from 5' to 3'. So, let me see if I can write this down. Somehow that makes sense in my mind so that I don't have to keep on freaking out every time someone asks me what direction something is going in. So what the hell is... I feel like something's like tickling my nose. Bruh. Okay, leading strand. I want to include DNA polymerase here. Because when someone says like it goes in the blank direction. It doesn't really tell me much because like I need to be more clear on what they mean by that. Like, are we following the template strand in that direction, or are we creating something in that direction? You know what I mean? It's like completely opposite meanings. So let's see. Let's see how this guy explains it.
It goes, this is the corresponding 5' end. So this and this are the same strand, and this one, if you follow it along, if you go all the way over here, is the same strand. So this is the 3' end and 3' end of it, and then this is the 5' end of it. Now, the first thing, and we've talked about this in previous videos where we did an overview of replication, is the general idea is that the two sides of our helix, the two DNA, the double helix needs to get split, and then we can build another side of the ladder on each of those two split ends. You can really view this as, if this is a zipper, you unzip it, and then you put new zippers on either end. But there's a lot of, in reality, it is far more complex than just saying, oh, let's open a zipper and put new zippers on it. It involves a whole bunch of enzymes and all sorts of things, and even in this diagram, we're not showing all of the different actors, but we're showing you the primary actors, at least the ones that you'll hear discussed when people talk about DNA replication. So the first thing that needs to happen, right over here, it's all tightly wound. So right there, it is tightly wound. And it actually turns out the more that we unwind it on one side, the more tightly wound it gets on this side. So in order for us to unzip the zipper, we need to have an enzyme that helps us unwind this tightly wound helix. And that enzyme is the topoisomerase. And the way that it actually works is it breaks up parts of the backbones temporarily so that it can unwind and then they get back together. But the general high level idea is it unwinds it. So then the helicase enzyme, and the helicase really doesn't look like this little triangle that's cutting things. These things are actually far more fascinating if you were to see the molecular structure of helicase. But what helicase is doing is it's breaking those hydrogen bonds between our nitrogenous bases. In this case, this is an adenine here, this is a thymine. It would break that hydrogen bond between these two. So first, you unwind it, then the helicase, the topoisomerase unwinds it, then the helicase breaks them up, and then we actually think about these two strands differently, because as I mentioned, you can only add nucleotides going from the 5' to the 3' direction. So
Okay. You can only add from the 5'. So we can only create the new strand from from its 5' to 3'. So, okay. Create new strand in 5' to 3'. Okay. Hopefully I will never be confused ever again. I probably will.
this strand
It's fine.
all are leading the strand. This one actually has it pretty straightforward. Remember,
Let's see.
So it can add going in that direction. It can add going in that direction right over here. This is the 5' to 3'. So what needs to happen here is, to start the process, you need an RNA primer. And the character that puts an RNA primer, that is DNA primase. We'll talk a little bit more about these characters up here on the lining strand. But they'll add an RNA, let me use some color you can see. An RNA primer will be added here. And then once there's a primer, then DNA polymerase can just start adding nucleotides. It can start adding nucleotides at the 3' end. And the reason why the leading...
Okay, so there is a common element in both of these routes, which happens to be both the DNA polymerase and the RNA primer. Okay. Oh, whoops. I forgot that this is not going to be there just yet. I need to include DNA primase in the lagging strand portion so that we can know exactly how it's going to attempt to create the same energy as the other strand. But in the opposite direction. Okay. There. Excellent. And then... I need way more space here. This is not that easy of a topic to just compress. Okay. And then that'll cover the leading strands situation. What we need to include then yes, okay, so this creates an Okazaki fragment that needs to be completed with the DNA ligase. Yes. Okay. Okazaki fragment. Ugh, that reminds me of an Apex map fragment. No, not again. Not the tilted towers. Okazaki fragment. Do not drop on fragment. Do not do it. DNA ligase. We can just let them finish explaining all that again.
This polymerase, and once again, they aren't in these perfect rectangles on this diagram. They're actually much more fascinating than that. You see the polymerase up there. You also see one over here. Polymerase. This polymerase can just, you can kind of think of it as following the open zipper, and then just keep adding nucleotides at the 3' end. And so this one seems pretty straightforward. Now, you might say, well, wouldn't it be easy if we could just add nucleotides at a 5' end? Because then we could say, well, this is going from 3' to 5'. Well, maybe that polymerase or different polymerase could just keep adding nucleotides like that, and then everything would be easy. Well, it turns out that that is not the case. You cannot add nucleotides at the 5' end. Let me be clear. This 3'.
Sad.
This strand over here. This strand right over here. This is the 3' end. This is the 5' end. And so you can't just keep adding nucleotides just like that. And so how does biology handle this? Well, it handles this
The most redundant way possible.
It'll add primers. And this diagram shows a primer is just one nucleotide. A primer is typically several nucleotides, roughly 10 nucleotides. So it'll add roughly 10 RNA nucleotides right over here, and that's done by the DNA primer.
Whoop,
The DNA primer is going along
whoop.
the
Make
lagging
a new
strand,
one. Make a new one.
the top strand, and it's
And then
adding
paste them all together.
the RNA primer is just one nucleotide. It tends to be several of them. And then once you have that RNA primer, then the polymerase can add in the 5' to 3' direction. It can add on the 3' end. So then it can just start adding. DNA like that. And so you can imagine this process. It kind of, you add the primase, put some primer here, and then you start building from the 5' to the 3' direction. You start building just like that, and then you skip a little bit, and then that happens again. So you end up with all these fragments of DNA, and those fragments are called Okazaki fragments. So it's Okazaki fragments. And so what you have happening here on the lagging strand, you can think of it as, why is it called the lagging strand? Well, you have to do it in this kind of, it feels like a suboptimal way, where you have to keep creating these Okazaki fragments as you follow this opening, and so it lags. It's going to be a
It
slower
feels like.
But then all of these strands can be put together using the DNA ligase.
I mean, it's not
only will the
that.
strands be put together, but then you also have the RNA being
No,
replicated
it's
with
pretty
the
suboptimal.
you're going to have a strand of DNA being replicated or being created right up here. And so when it's all done, you're going to have two double strands, one up here on the lagging strand and one down here on the leading strand.
Yay. Leading and lagging. Just like Miro while I'm trying to work on it. Oh boy. Come on. Oh, whatever. That'll work. Need to compress this to make it easier for me to... Oops. Don't want to group that. Or I can group it, but I'd rather group it visually. Like this. So now that is more in line with the lagging strand situation. Then now I can just go ahead and highlight it red. Because it's slow. And I don't like it. No. Just because it's slow. Alright, red. And then this will be green. We're going fast. Alright, nice. And then this can be yellow, which is like in between red and green, I think. Don't yell at me, I'm not a color theorist. Alright, so looks pretty good. Oh my god. There.
What we're going to do in this video is a little bit of a deep dive on transcription.
Ah yes, a deep dive into
just as
transcription.
bit of a review, we touched on it on the video
That's
on-
not good.
on replication, transcription, and translation.
Um,
Transcription in everyday language just means to rewrite something or to rewrite
I do
something
not
to
have the
a
space for
new
this.
form.
Okay. Time for some damage control. Let's see if I can do something about it. I was expecting us to like talk about chromosomes a lot more than we did. I think we'll get back to it, but for now I need to move this up. Get this stuff out of the way. RNA viruses can stay where they were. But I need way more space for transcription. Okay. I think this will do. Replication. Okay, maybe we can color the central dogma a certain color. Let's see, maybe purple. Oh god, you can't read that. Actually, you can change the opacity of this. I didn't know that. Wow. Oh, go figure. Okay. Why am I not dragging the... Okay. Stupid arrows are gonna get in the way and I can't change the color. 50%. And 50% again. Alright, nice. Let's figure
That's
out
essentially
what
what's
happens in
happening.
transcription.
Is when we take the information encoded in a gene in DNA and encode essentially that same information in mRNA. So transcription, we are going from DNA.
Years of study, my friend. Also, just like, it looks cooler than my notes. So I'd much rather remember this than have to remember my notes. I don't have a photographic memory by any means. If I did, then I would be able to memorize anything, whether in note form or mind map form. But at least with this, you can be like, okay, well, what kind of RNAs are there? Trying to imagine in my mind something branching off of RNA, ncRNA and tRNAs, and rRNAs. Basically, like, you know one spot on the map, and you can just try and imagine something else branching off of it, or branching towards it. Like, in this case, we can look over at RNA primers. What is that important to? What kind of thing uses that in its system? Replication. DNA replication. And so you're like, okay, how are they used in replication? Well, depends on what strand we're using, right? So there's going to be two branches, leading and lagging. And then we're like, okay, in the leading strand, it's just going to be the starting point. And if it's the starting point in the leading strand, then in the lagging strand, it must also function as a similar type of starting point. But because lagging strand has the unique property of being tedious as hell, we need to spam a ton of like RNA primers so that we have this list of things that we can build DNA from and then copy together, or not copy together, like glue together to make a proper DNA strand. So, I don't know. I think it makes sense. It helps. But yeah, certainly this would not be as easy to remember if I hadn't taken all those classes that I did. And again, even though I feel like I don't remember too much from my past classes, when I actually get to see the little hints, I can draw the map in my mind and arrive at some conclusion of what's going on in the system.
We are going from DNA,
even if it
to
might not be
messenger
as holistic as
RNA. And we're going gonna
people who are actually way smarter than me.
For proteins. So this first step is the transcription.
Yay,
DNA
transcription.
to messenger RNA. And then in a future video, we'll dig a little bit deeper into translation. We will translate that information into an.
Yeah, I'm not sure why I didn't put mRNA
Give a
here.
little bit of an overview of it. It's a little bit simpler in bacteria. You have the DNA just floating around in the cytosol. And so the transcription takes place. You start with that DNA, that protein coding gene in the DNA. And then from that, you code the messenger RNA. You see that in that purple color right over here. And then that messenger RNA can be involved with the ribosome. And
Maybe I should do mRNA here, and then mRNA is going to be approached by RNA. Give me that tool. There we are. Okay. So this is RNA, actually. And this will not be purple because I will not consider this as part of the central dogma, because the mRNA is specifically the RNA variation that we are using in this situation. At least for transcription. And then translation itself will require mRNA creating something else with the help of RNA. So, okay. We're good.
The translation process to actually
Let's
produce
go.
a polypeptide, to produce the protein. In eukaryotic cells, and we're going to get into a little bit more depth in this video, the transcription, the DNA to mRNA, that happens inside of the nucleus. And there's essentially two steps here. You go from DNA to what we would call pre-mRNA. Let me write that down. Pre-mRNA.
Oh my god, I forgot about it. Pre-mRNA. Pre-mRNA.
Call mRNA, which then can leave the
Bruh.
nucleus to be translated into a protein.
Because then
So
you
now
have
we
to
that
split
overview.
off all
Let's take a
the
look at this.
random
We're
shit
talking
that
about a
needs
eukaryotic
to
cell,
be
but processing might actually go on.
deleted.
Right over here, we are going to
Okay.
start with the protein coding gene inside of the DNA, right over here. And we are going to, the primary actor that's not the DNA or the mRNA here is going to be RNA polymerase. It's used to create a sequence that will become a nucleotide sequence that will become the messenger RNA. So this RNA polymerase, it needs to know where to start. And the way it knows where to start is it attaches to a sequence of the DNA known as a promoter. And every gene is going to have a promoter associated with it, especially if we're talking about eukaryotic cells. Sometimes you might have a promoter associated with a collection
That's
of
right.
genes as well. But in general, if you've got a gene, you're going to have a promoter. So
Okay.
that's how the RNA polymerase.
Let's see here. Ignition sites included in the promoter. Okay, so we'll make a promoter bubble. Do I want to keep this purple? That is the question. I mean, really the central dogma is just the DNA, RNA, protein part. But I think all the steps in between can be considered as part of that. So let's keep it as purple for now. And see if I can make this prettier. Promoter, which includes recognition sites, which is not purple, because that is just a part of that. It is not included in the actual system that I'm going to create, this circuit. In fact, I'm not even sure how important this is, because obviously it should have recognition sites so that the polymerase can even show up to begin with. But, you know, just going to go for completeness. Okay. Right, I am getting way ahead of myself by putting pre-mRNA here. Let's put RNA polymerase down. And then we can worry about introns and extrons. Yeah, exons. They don't have the tron part. Unfortunate. And it's been forever since I've watched the Tron remake. Never watched the first one, but I don't think anyone, well, anyone from my generation has watched that original one. The Tron remake was epic. Okay, I have once again gotten ahead of myself by accident. Don't even worry about it.
Polymerase knows to attach right over there. And so once it attaches, well then it is able to separate the strands. It separates the strands. And it's pretty interesting because when we went deep into replication, you saw all of these actors, the helicase and whatever else, but this RNA polymerase complex is actually quite capable. Not only it separates the strand, and then it's actually able to code for the RNA. And it does that the same way that when we study DNA polymerase, it does it in only one direction. It can only add more nucleotides on the 3' end. So it encodes from the 5' to the 3' direction. Notice this arrow here, we're extending it on the 3' end of the RNA. And so as you can see here, when it does this, it's only encoding one side of, or it's only interacting, I guess you could say, or coding complementary information to one side. Let's think about this a little bit. We could call the side that it is interacting with, you call that the template strand, because that side of the DNA is acting as a template for forming that RNA. But if you think about the information that RNA is actually going to encode, well, it's going to contain the same information as the coding strand of DNA, as the other strand of DNA, because these nucleotides right over here, this nucleotide is going to be complementary to this one over here, just as this nucleotide was complementary to that.
I mean, okay. So coding and template strands are pretty intuitive, at least on my end. Once again, situation of completeness. Let's just include them. Coding and template. Now I know this need for completeness is slowing us down. I am on the clock here. That's why I need to do this stream very often. But just looks nicer with more stuff.
That one over there. And you can see it in a little bit more depth if we actually were to add the nucleotides. So this is the template strand. If you have thymine, well, on the RNA, you'd have the adenine. And look, on the coding strand of DNA, the one up here, you would also have an adenine. And they are essentially, the coding strand and the RNA essentially end up being the same sequence, with the one difference is that you won't find the thymine in the RNA. Instead, you'll find a similar nitrogenous base, and that is uracil. But uracil plays the role of thymine, so you're essentially coding the same information. So
Bro,
once again, this
thymine is such a rebel.
but it's going to be
Doesn't want to join in on RNA.
It's essentially going to have the same information that we have in the coding strand.
You have to
Just
find a
appreciation of what this
replacement.
looks like, and I would even write,
Bruh.
in quotation, I even did little quote things with my fingers when I said that, is that
What's up with
it's hard
that?
to really visualize what these things look like. But you can see here that the RNA polymerase complex, and this is for a specific organism, can be very, very complex and involved, and it's fascinating how these things interact. And every time you're studying biology, and someone like me is going to give you these nice, clean narratives of how these enzymes interact with the different macromolecules like the DNA or the RNA, you should always remember, this is amazing. These are these molecules interacting with each other, bouncing into each other. It's happening incredibly fast inside of the cell. You should be in awe of this. It's happening in all of your cells, or as we speak. So this is pretty incredible stuff. So the next thing you have to think about, this right over here, we are extending the RNA. Well, when does this thing actually stop? And it stops once we, so this RNA polymerase is going to keep going on, and then it's blue where we've labeled this a terminator. So let me write.
DNA ligase nuts. Amazing.
And there's multiple ways that signals to the RNA polymerase that, hey, it's time to stop. Or more particularly, that it somehow creates something structurally that the polymerase...
It's time to stop.
Right here is that the mRNA that's coded, and this is typical, or this can happen in bacteria, is that the mRNA that's coded forms a hairpin. So it has to have the right complementary base pairs right over here to form this hairpin. But this hairpin, along with the things around the hairpin, essentially impair the polymerase to keep on going. And so the complex kind of changes a little bit, and so it lets go, or at least that's how people believe it. There's other forms of how the terminator can act. It might be sequences that parts of the polymerase complex recognize, and it makes a conformation change. So the RNA polymerase lets go. Now we're talking about a prokaryote. We're done. We would have formed, this would be our messenger RNA, which then can go to a ribosome and then be translated into a protein. But if we're talking about a eukaryote, well, then we have to do a little bit of processing. If we're talking about a eukaryote, if this is a prokaryote right over here, this would be our mRNA. If this is our eukaryote,
Post-processing is killing me, man.
Which now has to be processed. And you might say, well, how is that going to be processed? Well, there's
Also, I just realized that you were making a JoJo reference to
the 5' cap. This is a modified
the episode that we were literally just watching yesterday. I couldn't read the bottom of your message because OBS likes to be a prick. What am I doing again? So wait. RNA polymerase, I want to include the terminator. Arnie is here, and he is making sure that our RNA is as beautiful and pure as possible. Amazing, so that we can process it and get rid of all of the exons and introns. Okay, the RNA, yes, so we can now include
guanine, modified guanine right over here, which is going to help in the translation process as the ribosomes attach onto it. And then you have this poly A tail, and it's called a poly A tail because it has a bunch of adenines at the end right over here. And these not only help in the translation process, it helps make sure that the information is more robust, that the ends of the mRNA don't in some way become, or makes it less likely that they're going to become damaged. Now the other thing that needs to be processed, and this is one of those fascinating things in evolutionary biology, is that we will have in this mRNA sequence, you're going to have parts of the sequence which we currently consider to be nonsense sequences, and we call them introns. And I'm going to put it in quotes because in general in evolution, it's seldom that things have absolutely no purpose, but these are not coding for the protein that is going to be coded by our initial initial gene. And so these are actually processed out. They are spliced out. I'm not going to go into all the details of the actors that cause the splicing, but as part of this eukaryotic processing, you add the cap, you add the tail, and then you splice out the introns. And then once you splice out the introns, all you have.
Ugh. Okay. All A tail, and then we have the introns and extrons. Okay. And then we take out the introns, right? So that would mean we get hella exons. And then those get added together. Ugh, it's so hot where I am right now. Feel totally gross, bruh. Gonna have to take a shower. Oh well. Exons, let's get this done first. Exons. Here. Who needs hygiene? Studying is forever. Amazing. And they thought, and y'all thought that gamers were the only ones that don't shower, bruh. Okay. Let's see, RNA is supposed to be branching off of RNA. Come on now. Okay, exons. We want to delete the introns, maintain the exons, so this can actually stay purple.
Left are the exons. So you have that, it's going to be connected to that, it's going to be connected to that, and so this is what you have resulted. This is in a eukaryote, you'll have this mature mRNA, and that's what we saw. That's what we saw right over here. That can then, let me underline that in the color you can see, right over here, which then migrates out of the nucleus to a ribosome where it can be translated. In the earlier video on DNA replication, we go into some detail about leading strands and lagging strands and all of the different actors, all of these different enzymatic actors. But I left out what is probably the most mind-boggling aspect of all of this, and that's the speed and the precision with which this is actually happening. As we talked about in that video, it feels pretty complex. You have this topoisomerase that's unwinding things, the helicase is unzipping it, and then you have the polymerase that can only go from the 5' to 3' direction. It needs a little primer to get started, but then it starts adding the nucleotides. On the lagging strand, you have to have you get the RNA primer, but then it's going from, once again, from 5' to 3'. So you have these Okazaki fragments, and all of this craziness that's happening. Remember, these things don't have brains. These aren't computers. They don't know exactly where to go. It's all because of the chemistry. They're all bumping into each other and reacting in just the right way to make this incredible thing happen. Now, what I'm about to tell you is really going to boggle your mind because this is happening incredibly fast. DNA polymerase has been clocked,
Boggle your
mind. Approaching 1,000 base pairs per second. I think the number that I saw was 700 something base pairs per second. So polymerase, let me write this down. This is worth writing down.
That's pretty fast.
It gives you a sense of just how amazing what the machinery in your cells are. So it's been as high as, and it can change, it can speed up and slow down, and that's actually been observed. But polymerase as fast as 700 plus base pairs per second. So if this on this diagram, man, it's just zipping along, at least from our perceptual frame of reference. So a second seems like a very short amount of time to us, but on a molecular scale, these things are just bouncing around and just getting this stuff done. Now, the second thing that you might be wondering, okay, this is happening fast, but surely it has a lot of errors. Well, the first thing you might say, well, if it had a lot of errors, that would really not be good for biology because you have DNA replicating all throughout our lives, and at some point you just have so many errors that the cells wouldn't function anymore. And so lucky for us that this is actually a fairly precise process. Even in the first pass of the polymerase, you have one mistake. Let me write this down because it's amazing. One mistake for every approximately 10 to the 7th. So this is 10 million nucleotides. And that might seem pretty accurate, but you got to remember we have billions of nucleotides in our DNA, so this would still introduce a lot of errors. But then there's proofreading that goes back and makes sure that those errors don't stick around. And so once all the proofreading takes place, it actually becomes one mistake for every approximately 10 to the 9th nucleotide. So approximately you can reduce it at an incredibly fast pace, as fast as 700 plus, approaching 1,000 base pairs per second, and you have one error every billion nucleotides, especially after you go through these proofreading steps. And so it's incredibly fast and it's incredibly precise. So hopefully that gives you a better appreciation for just the magic. That's literally, look at your hand, or just think about this is happening in all of the cells or most of the cells of your body as we speak.
I like how that was just a DNA polymerase appreciation video. Yeah, I don't think I need to know that specifically. I know that it's fast, and I know it's precise, because we have proofreading. Because if something goes wrong, it goes really wrong. Or it can go really wrong. Especially if we have a frame shift. That gets real awkward real fast.
are made up of really long strands of DNA all wound up into our
Oh, no.
cells. So something like, well, I'm just kind of drawing it as a random long strand of DNA all wound up in itself. And on that strand, you have sequences, which we call genes. So that might be one gene right over there. This might be another gene. That might be a gene right over there. And each of those genes can code for specific polypeptides or specific proteins. And
Yeah, I'm gonna need to make more space again. Let's actually move this out to the side. Maybe. Okay. And look at that. Look at the precision. Amazing. Okay. Now I can place this here. It remains attached. We have all this space. Viruses. RNA viruses are pretty cool. I do want to include that somehow. Keep it simple. Nice. All right. Moving in for the kill.
Information encoded in these genes into
mRNA
DNA.
time.
How do you go from that? How do you go from the gene, which is encoded in DNA? How do you go from that to protein, which is made up of polypeptides, which are made up of amino acids? And this is often called the central dogma of biology. But we already saw in the video of transcription that the first step is to go from the gene to messenger RNA. That the RNA, the messenger RNA, you can do it as a transcript. We have rewritten the information now as RNA. And then the next step, which we're going to dive into in this video, is going from that messenger RNA to protein. And this process is called translation, because we're literally translating that information into a polypeptide sequence. And you can see a little bit visually here. This is all review. We covered a lot of this in the video on transcription. And the overview video on transcription and translation is if you look at a eukaryotic cell and the bacteria in a prokaryotic cell, it's analogous. You just don't have the nuclear membrane, and you're not going to do the processing step that I'm going to talk about in a little bit. And we went in detail on the video on transcription. But you start with the DNA. You have your RNA polymerase as the main actor that's able to transcribe the RNA from that. If we're talking about a eukaryotic cell, what you end up with, we wouldn't call mRNA, we would call that it's pre-mRNA. Pre-mRNA, which then it needs to be processed. The introns need to be taken out. We add a cap and a tail here, and if we're talking about a eukaryotic cell, we'll then formally call that mRNA, and then it can travel, and this is where we get into the translation step, it can travel to a ribosome, which is where it will be translated into a polypeptide sequence. And you see the analogous thing happening here in this bacterial, or this prokaryotic cell right over here, because you don't see the nuclear membrane because it's prokaryotic, and you don't see that processing step. So you can just consider this straight. This is mRNA right over there. So the questions are, well, how does this thing happen? What even is a ribosome?
Wait a minute.
So let's zoom in a little bit.
Oh, shoot. The poly A tail is on the mRNA. Like the actual mRNA, the mature one, not the exons, or the pre-mRNA one. Okay, let's do that real quick. How will I represent that? Well, I guess we can just keep it simple. Like that, and that. Obviously, it's not going to be red, because that wouldn't make sense. All right, nice.
On a ribosome right over here, and there's a couple of interesting actors. One, as you can imagine, is the ribosome itself, and it is made up of proteins plus ribosomal RNA. So in the video on transcription, we're already familiar with messenger RNA, and we often view RNA, like DNA, as primarily encoding information. It's acting as a transcript for a gene, but it doesn't have to only encode information. It can also, so it's proteins plus, that's not a T, this is a plus. It can also provide a functional structural role, which it does in ribosomal RNA. And this big, this looks like an oversized hamburger bun or something right over here. This is a super oversimplification of what a ribosome looks like. I encourage you to do a web search for image searches for ribosomes, and you can get a more appreciation of how beautiful these structures are and how intricate they actually are. So this is the site, and you can broadly think of the ribosome as having this, this is the top bun and the bottom bun, and it's going to travel along the mRNA from the 5' end to the 3' end.
Oh my god, it's a hamburger.
and turning it into a sequence of amino acids.
Okay. Man, I did not give myself enough room here. Let's move that a little. Okay. What the hell is going on with this? Oh my goodness. This branch is not cooperating. Okay. All right. Nice. It's a little bit more compact. What I can do instead is actually make this align here. So now, boom, done. All right. Let's see. I can translate using a ribosome.
So how does that actually happen? Well, each of these every three nucleotides there, we call that a codon. So that's a codon. This is in a color that is visible on both white and black. So this next three nucleotides is a codon. This is a codon. This is a codon. And what's actually the information is actually encoded in the nitrogenous bases. So this first codon right over here, we see it's AUG. So the nitrogenous bases are adenine, uracil, and guanine. And this codon, it codes for the amino acid methionine. But this is a good one to know. AUG. Let me write it over here. AUG is known as the start codon. Start codon. This is where the ribosome will initially attach to start translating that messenger RNA. And so the way that this drawing is that we are just starting to translate this messenger RNA. So how does that actually happen? How do we get from these three-letter sequences to specific amino acids? Well, let's think about it. How many possible three-letter sequences are there? Well, there are 4 possible nitrogenous bases there. So there's 4 possible. So if you have a codon and it has three places, there's 4 possible things that could be in the first place. There's 4 possible things that could be in the second place, and there's 4 possible things that could be in the third place. So there are 64 possible.
Okay, whoa, my friend. I need to move proteins right now. Get a bit more space. Okay. Then we know that RNA is going to be included. rRNA can be included in the creation of the ribosome, whereas tRNA is going to be matched with mRNA in order to produce the next protein by matching the amino acids together, and then we'll be golden. So let's see, tRNA. And then I want to tell that this is going to a codon trio. It's amazing that they actually delayed this game for this song. Good decision. Oh no. Is there really no way to micromanage where these bubbles go? I'm so disappointed in that. Ugh. Whatever.
Permutations. 4 times 4 times 4 permutations. So you can think of it, there's 64 different codons, different ways of arranging the A, the U, and the G. And that's good because there are many amino acids. And this is actually overkill because there's actually 22 standard amino acids. 22 standard amino amino acids, and 21 that are typically found in eukaryotic cells. So we have more than enough permutations to cover the different amino acids. And it's not hard to find tables that will actually show us what the different sequences, what they actually code for. So you can see here, you can take the first letter, the second letter, and then the third letter. Look at the different sequences, and you say, okay, look at that. AUG, adenine, uracil, guanine, that codes for methionine, right over here. You could do that with any. You could say cytosine, uracil, that codes for leucine. And you can see that it's not just one amino acid per codon. But here you have 4 codons, all code for leucine. And so it turns out that 61 of the codons, let me write this down. So 61 of the codons, of the possible 64, code for amino acids.
I feel like I should represent thymine becoming uracil. Well, not really becoming, sort of, being replaced by uracil in the RNA production line. Thymine becomes uracil. I feel like it's more accurate to say that adenine produces or encodes for uracil to be matched with it. But anyway, let's go with that.
Amino acids. And 3 play a role that essentially tells the ribosome to stop. 3 codons are stop codons, and you can see them right over here. UAA, UAG, UGA. That's how the ribosome knows to stop translating. So AUG, that's a start codon, and it codes for methionine. So that lets you know that, well, these polypeptide chains are going to start with methionine, and then these characters tell it where to stop. But how does the amino acid actually how do they all get tied up together to form this polypeptide? And how do they get matched up? How do they actually get matched up with the appropriate codon? And that's where we have another RNA-based actor, and this is tRNA. So tRNA, the T stands for transfer RNA. There's a bunch of different tRNAs that each can bind to specific amino acids, and on parts of those tRNA, they have what are called anticodons that pair with the appropriate codon. So this tRNA, and that's not what it looks like, I'll show you in a second what it looks like. That's a tRNA molecule, tRNA. At one end of the molecule, it's binding to the appropriate amino acid, methionine, right over here. And then at the other end of the molecule, although it's in the middle of the tRNA actual chain, you have your anticodon. And your anticodon matches up to the appropriate codon. And so this is how they bump into each other the right way, and the ribosome is going to facilitate it, that the AUG is going to be associated with the methionine. And if we look at what tRNA actually looks like, and this is still just a visualization, so this is a strand of tRNA, you get a sense of, okay, it's a sequence of RNA right over here. This is its, I guess you can think of its two-dimensional structure, but then it wraps around itself to form this fairly complex molecule. And the anticodon, which is right here, it's kind of in the middle of the sequence, it forms the basis for this end of the molecule. That's the part that's going to pair with the codon on the mRNA. And then at the other end of the molecule, is where you actually bind to the appropriate amino acid. So I know what you're thinking. All right, I see that the ribosome, it knows where to start. It starts at the start codon. I see how the appropriate tRNA can bring the appropriate amino acid, but how does the chain actually form? And you can do this in 3 steps, and associated with those 3 steps are 3 sites on the ribosome. And the 3 sites, we call this the A site. I'm not going to be able to see if I write it in black. A, or yellow. All right, let me write it in blue. So that is the A site. This is the P site. And this is the E site. And I'll talk in a second why we call them A, P, and E. So the A site is where the appropriate tRNA initially bounds, the tRNA that's bound to an amino acid. And so you can see we're starting the translation process. The next thing...
Okay, so in order to actually include the A, P, and E sites, let's put them in between the codon and the tRNA. A site, E site. And then E site. Not to be confused with E-scored. All right, let's see here. Amazing.
What's going to happen is another tRNA, the one that has an anticodon that matches the UAU, that's going to bond over here on the A site, and it's bringing the appropriate amino acid with it. It's bringing the tyrosine with it. So why is it called the A site? Well, A stands for aminoacyl. You can do, or an easy way to remember it, it's the tRNA, it's the place where the tRNA that's bound to an amino acid, just one amino acid, is going to bind on the ribosome. And so once that happens, once this character comes here, let me draw that. Once this character comes right over here, it's going to be AUA, and it's bound to the tyrosine. Well, then you can have a peptide bond form between the 2 amino acids, and the ribosome itself can move to the right. So this tRNA will then be in the E site, this tRNA will then be in the P site, and then the A site will be open for another amino acid carrying tRNA. So what does P and E site, what do the P and E sites stand for? Well, you can see a little bit more clearly right over here. So the P site is where you have the polypeptide chain actually forming. And so the P site is often, well, one way to remember it is that's where you have the polypeptide chain, and now you have a new A site where you can bring in a new amino acid, and then the ribosome is going to shift, once this is bound, the ribosome, the peptide bond forms, and then the ribosome can shift to the right. When the ribosome shifts to the right, we're going to be in this position, where the thing that was here, that was in the A site, now the polypeptide is attached to it. It is going to now be in the P site, and the thing that was in the P site is now going to be in the E site. It is now ready to exit, and that's why it's called the E site, because that's the site from which you exit. And so this is going to keep happening until we get to one of the stop codons. And when you get to one of the stop codons, then the appropriate polypeptide is going to be released, and we will have created this thing that could either be a protein or part of a protein. So this is very exciting, because this is happening in your cells as we speak. This is, and in fact, if you think about things like antibiotics, the way that they work is that, or the way that antibiotics work is that ribosomes in prokaryotes are different enough than ribosomes in plants and animals or in eukaryotes that we can find molecules that hurt the function of ribosomes in prokaryotes, but don't do it to eukaryotes. And so if you have bacteria in your bloodstream, and if you take the appropriate antibiotic, it can disrupt this translation process in the bacteria, but not in your cells that you want to keep.
There you go. Let's see. How long is this?
Let's
6 minutes?
talk about some of the
Sure.
differences between how translation happens in prokaryotic cells and how it happens in eukaryotic cells. And I want to focus mainly on the mRNA just
Oh
before
my
it's actually
lord, it's
translated.
loud.
So let's start with our prokaryotic mRNA, and let's look at our 5' side first. So we have this yellow part right here, and that's the non-coding region. And it's called the non-coding region because the ribosome is not actually going to read that part. So that particular sequence of amino acid is not that important. And then after the non-coding region, we have the
Okay, wait, hold on. Where even are we on the, like, list of topics? Oh my lord. I don't think we finished DNA, did we? Holy crap. I guess I'm not that surprised. DNA is a very complicated subject. Okay. Well, I do have a lot of work today, so we're going to leave it as is today and come back to it tomorrow. All right. I'll see you guys a little later. And good luck with your studies. And that's it. Goodbye.